Hello everyone, welcome to the Tim Booker channel. Wishing you all an enjoyable listening experience. Today, we're going to discuss the book Brainscapes. If you closely examine the human brain, you'll find that it's spherical in shape with many deep grooves on its surface, separated by raised gray areas, resembling a wrinkled walnut. While it may not be aesthetically pleasing on the outside, the brain harbors intricate maps within. It's through these maps that we perceive our surroundings, hear words spoken to us, touch objects with our hands, and imagine the future. The brain is, in fact, a rich atlas, not a romantic metaphor but an objective physiological structure. Each so-called map corresponds to specific regions in the brain, responsible for helping us process and understand the world around us. Unlike conventional geographic maps, the brain's maps are not composed of dots and lines, their basic units are neurons. In the brain, neighboring neurons often connect and share similar functions. When we visualize these functional areas, we can see how the brain's maps are distributed within our heads. So, what exactly are these brain maps? By definition, maps are spatial representations of other things, and brain maps are naturally spatial representations of various aspects within the brain, including sensory perception like vision, hearing, and touch, as well as abilities like movement, intention, and abstract thinking. In essence, brain maps are visual representations of different brain regions responsible for distinct functions. For example, a region at the back of the brain is responsible for processing visual information, hence we call it the visual map. This terminology is both easy to understand and vivid, aiding our comprehension of brain function. Brain mapping falls under the realm of neuroscience. The author of this book, Rebecca Schwarzlos, is a professional in this field. She is a neuroscientist at Washington University in St. Louis, USA, with a doctoral degree in neuroscience from the Massachusetts Institute of Technology. She has also served as the chief editor of the academic journal, Trends in Cognitive Sciences. Brain mapping is one of her areas of expertise. In this book, Schwarzlos demonstrates that brain maps are an inevitable outcome of evolution, allowing us to efficiently process external information at a lower cost. The formation of brain maps is regulated by both genes and environmental factors. Life experiences, particularly early ones, significantly shape the layout of brain maps, profoundly influencing the cognitive abilities of each individual. The subtitle of this book poses a question: why do we remember, imagine, learn, think, and act in the ways we do? The answer is clear: brain maps determine all of these aspects. Since brain maps are so crucial, I will take you through two sections to provide a detailed understanding. The first section will discuss the usefulness and significance of brain maps through various examples, while the second section will delve into the development of brain maps and how we can optimize their service to us. Let's discuss the utility of brain maps and their significance through a few examples. Our understanding of brain maps actually originated from an unexpected event during a war. In 1904, the brutal Russo-Japanese War erupted over territorial and naval dominance. In this war, Russian soldiers wielded the new Mosin-Nagant M91 rifle, known for its high firing rate and long range. Bullets fired from this rifle often passed through the brain and skull, leaving neatly edged bullet holes upon impact with the head. Surprisingly, these injuries didn't cause much harm to the brain's internal structures. Many Japanese soldiers who were shot in the head survived, which inadvertently increased the survival rate of soldiers with head injuries. However, some soldiers who were shot in the back of the head noticed visual problems despite their eyes remaining unharmed. They experienced a hollow-like blind spot in their field of vision, varying in size and moving with their gaze. A young ophthalmologist named Tatsuji Inoue keenly realized that this phenomenon likely held secrets about the connection between the brain and vision. In fact, in the late 19th century, just before the outbreak of the Russo-Japanese War, the scientific community had already identified that the part of the brain responsible for processing visual information was located near the back of the brain. However, due to a lack of sufficient research samples, the exact location remained unknown. Inoue was fortunate, as the casualties of the Russo-Japanese War provided him with enough subjects for observation. After examining the blind spots in each soldier and the locations of their head wounds, he detailed his findings in a paper. He revealed that there exists a visual map within the human brain. This map is divided into two halves, symmetrically distributed in the rear regions of the left and right hemispheres of the brain. Compared to the corresponding visual field, the visual map is inverted both vertically and horizontally. The right visual field is represented in the left brain, and the left visual field is represented in the right brain. The lower part of the visual field corresponds to the upper part of the visual map, and the upper part of the visual field corresponds to the lower part of the visual map. Moreover, this visual map is heavily distorted, with a disproportionately large representation of the central visual field, similar to placing a magnifying glass over the central visual area. For example, when you stand in front of the Mona Lisa, gazing at her smile, her face occupies the majority of your visual map, while the surrounding view occupies a small area. By simply using your imagination, you can sense that this distorted map is quite peculiar. But why is it constructed in such a distorted manner? The reason is closely related to the purpose of brain maps. In simple terms, the brain optimizes its resources for the most critical tasks. Think of the brain like a microchip. The more transistors it has, the more powerful it is. Similarly, the more neurons in the brain, the smarter it becomes. The brain volume of cows and chimpanzees is similar, but chimpanzee brains have more densely packed neurons, and their total neuron count is higher, making them much more intelligent. Having more neurons is beneficial because they enable organisms to perform more complex and precise actions. They also support abilities like navigation, memory, self-control, planning, and reasoning, all of which are advantageous for survival. However, having too many neurons comes at a cost. Neurons are energy hungry, with each pulse they send requiring a significant amount of energy to transmit specific molecules. The brain, despite comprising only 2% of body weight, consumes 22% of the body's energy, imposing a substantial energy burden. Additionally, neurons take up space. Each neuron needs to connect directly with hundreds of other neurons in the brain to fully function, and these connections, both vertical and horizontal, occupy physical space. It's easy to imagine that if there were too many neurons in the brain, it simply wouldn't fit. Neurons are both energy-consuming and space-consuming, so more isn't always better. Animals must balance their neuron count according to their actual needs. Furthermore, to save space occupied by vertical and horizontal connections, neurons with similar functions in the brain generally cluster together. For instance, neurons responsible for vision are close to each other, those responsible for central vision are even closer, and neurons responsible for a specific point within central vision are closely adjacent. It's not difficult to imagine that when neurons corresponding to every visual point are located close to each other, the brain naturally projects the outline of the visual map. However, outlining the visual map is not enough to fully utilize it. If you've ever noticed, within the entire field of vision, only the central vision is clear, while the peripheral vision is relatively blurry. This phenomenon aligns with the structure of our retinas. The central region of the retina is called the fovea, where light receptors are much denser than in other areas. Therefore, the brain needs to allocate more neurons to process visual signals from the fovea. Consequently, the visual map must undergo further distortion to meet the demands of processing visual information. At this point, you might be wondering, why can't we make the entire field of vision clear? The answer lies not in unwillingness but in impossibility. To make the peripheral vision as clear as the central vision, the area occupied by the visual map would have to increase by 13 times. Our brains are precious real estate, and each region has its own function. It clearly doesn't have that much space to spare. So, the brain opts for a compromise, preserving the central vision while relatively neglecting the peripheral vision. It achieves this through rapid eye movements and constant scanning, ensuring that we can promptly perceive useful information. By introducing the concept of the visual map, you should now understand that brain maps fundamentally represent the brain's synthesis of resources to accomplish its most crucial tasks. Their purpose is to allow us to make the most of our limited brain resources, processing external information as much as possible and as effectively as possible to meet the challenges of survival. It's worth mentioning that even with brain maps, we still can't handle this busy world at all times. Brain maps must work in conjunction with the use of attention to truly fulfill their mission. For instance, when you are attentively listening to this book program, you might momentarily disregard people passing by or the sensations your shoes provide to your feet. When you shift your attention to the sensation of your shoes upon hearing this sentence, the audio in your ears remains unchanged, and the sensation in your feet remains the same. However, the change in attention significantly alters the perception of the two maps in your brain. Attention, too, is a compromise between brain resources and efficiency. Although it cannot achieve perfection, it is undoubtedly an evolutionarily effective compromise. We've been using the visual map as an example, but the brain contains many other maps. For instance, in the cerebral cortex, there is a long, narrow region extending from the back of the head to the top, known as the primary somatosensory cortex. Neurons in this region make up the brain's tactile map. Different locations within the tactile map correspond to the tactile sensations of various body parts, ranging from the mouth, tongue, lips, face, thumb, fingers, palm, forearm, torso, pelvis, legs, and feet. Once sensory receptors in these body parts receive signals, they transmit them to the corresponding regions in the tactile map. The tactile map processes these signals, allowing us to perceive tactile sensations. Similar to the visual map, the area of the tactile map corresponding to a body part becomes more substantial, the more sensitive the tactile sensation of that body part. For instance, the area corresponding to the fingertips in the tactile map is large, making fingertip sensation highly sensitive, while the area corresponding to the torso is smaller, resulting in less acute tactile perception. The disproportionality between the surface area of body parts and their corresponding areas in the tactile map highlights the significant distortion inherent in this map. For other animals, the logic of distorting brain maps based on their needs is the same. Selective distortion of brain maps is a common strategy. For example, a mouse's whiskers occupy over a quarter of its entire tactile map, while a pig's nose occupies over half of its tactile map. The more critical the body part, the larger the corresponding area in the tactile map, and the more pronounced the distortion. In essence, regardless of the animal, this logic of distortion in brain maps remains consistent. Some tasks can be accomplished with a single brain map, while others require multiple maps to work in coordination. For instance, we can learn mathematics and perform mathematical calculations because we flexibly utilize multiple maps. In the top of the human brain, there are simple number maps. These number maps allow us to understand relatively small numbers, such as integers up to 6. However, when it comes to comprehending larger numbers, the existing number maps fall short. So, how does the brain address this issue? It does so in a brilliant way. One approach is to associate numbers with spatial locations. For example, the left side of space might represent smaller numbers, while the right side represents larger numbers. When comparing two number sizes, we instinctively arrange them in space, with the left number being smaller. This description simplifies mathematical thinking significantly, but it is enough to demonstrate the brain's flexibility. The brain can appropriate multiple maps as needed, using a few basic abilities to flexibly assemble numerous complex capabilities. In this way, it can infinitely expand its cognitive territory without increasing the size of the skull. The significance of brain maps is once again greatly extended. They not only provide us with fundamental thinking tools but also allow us to flexibly assemble these tools. The seeds of wisdom are sown on the territory that the brain maps hold. Alright, we've discussed the uses and significance of brain maps. Now, let's delve into how they develop and how we can make them serve us better. The brain, composed of over 100 billion cells, sculpts these intricate maps within its incredibly complex structure, akin to a super project. However, simplifying it, the development of brain maps essentially consists of two steps. The first step involves constructing the framework based on genetics. Brain map development begins early in embryonic stages, guided by various chemical substances. Newly born neurons migrate to designated positions under this guidance, laying the foundation for the adult brain's basic blueprint, specifying the relative size and location of each brain map. Let's take the visual map as an example. Once the nerve cells responsible for transmitting signals from the eyeballs to the brain establish themselves, they extend long axons. These axons continuously explore and move forward. During this time, chemicals secreted by the brain instruct these axons on where to grow and guide them from the original eye location to an area in the brain called the thalamus, where they precisely connect with thalamic neurons. Subsequently, neurons in the thalamus responsible for processing visual information also grow axons, moving towards the primary visual cortex and establishing connections there. Through this process, the framework of the visual map is constructed in the primary visual cortex. However, having a framework alone isn't sufficient. The neurons forming the visual map need to cooperate to function effectively. During development, retinal cells spontaneously generate waves of neural activity, transmitting between thalamic neurons and primary visual cortex neurons, thus enhancing their coordination. The development of the auditory map follows a similar pattern. The auditory map's role is to convert sound signals received by the ears into the specific sounds we hear. This allows the neurons within the auditory map to function normally. After the initial framework of the auditory map is established, a specific structure within the fetal ear generates neural activity waves spontaneously. These waves are then sent to the thalamus and auditory map, training the neurons in these areas. In contrast, the development of the tactile map is more intriguing. Because the fetus is immersed in amniotic fluid, its skin is smooth, and it's challenging for tactile sensors to form coordinated neural activity waves based on touch sensations. Therefore, the natural movements of the fetus, such as kicking and random limb movements, induced by the amniotic fluid, stimulate tactile sensors. These sensors generate waves, which are first sent to the thalamus and then to the primary somatosensory cortex, thus refining the developing tactile map. Mothers often feel their babies kicking during pregnancy, indicating that these little ones are in the process of forming their tactile maps. In the brain, the tactile map is closely adjacent to the motor map. The motor map controls body posture, and there is a close relationship between the tactile and motor maps. Neural activity waves from fetal tactile sensors, after passing through the tactile map, further propagate into the motor map, training motor map coordination, providing a twofold benefit. By the late embryonic stage, the primary connections in the brain have been successfully established. Although the fetus hasn't seen the world yet, it already possesses the basic framework of brain maps. Now, let's look at the second step in the formation of brain maps, which involves referencing the environment and rearranging. After birth, brain maps are far from being fully formed. In early life, brain maps are highly susceptible to environmental influences. Newborns continuously gather information from the external world and adjust their growth trajectories based on the signals they receive. Research has shown that by enhancing a newborn's learning in specific areas, brain maps can adjust resource allocation, leading to the growth of more connections within the corresponding brain map. This results in an increase in the thickness of the region where these brain maps reside. This phenomenon has been experimentally verified several times. For example, simply playing 3 hours of a mother's heartbeat to premature infants in the neonatal ward, or sounds that could be heard in the womb, gradually thickens the auditory cortex of the infants. Similarly, frequent stimulation of an infant's toes can lead to an increase in the thickness of the region in the tactile map corresponding to the toes. However, these adjustments mainly occur in the early stages of a person's life. As one grows older, the degree of adjustment decreases. For instance, some infants are born with congenital cataracts, rendering their retinal cells unable to receive visual signals from the external world. This disruption severely affects the development of the visual map. If the cataracts are removed within a few weeks of birth, there is a high probability of restoring normal vision. At this stage, the brain maps still possess a high degree of adjustment flexibility. Each week of delay in providing visual stimulus reduces the chances of a child recovering normal vision. If there is a prolonged absence of visual stimulation, the functionality of the visual map will eventually degrade. But why does this happen? Can't the brain keep these functions indefinitely? In fact, this is an adaptive strategy of the brain to ensure that no part of it is wasted. For instance, some children are born blind, and their vision cannot be corrected through surgery, condemning them to a life in darkness. In such cases, the visual cortex and complex visual maps in their brains, which would otherwise go unused, are repurposed. Over time, the visual cortex in blind children is reassigned to new functions, such as participating in language processing as part of the language map. In more extreme situations, the brain can even reconfigure all brain maps with half the volume. For example, researchers once studied an infant with a rare condition where the right hemisphere of the brain stopped growing early in development, leaving essentially only the left half of the brain. However, apart from minor difficulties in the left side of the body's movements, all other functions were entirely normal. For instance, despite having only half the visual map, this individual's vision was indistinguishable from that of a typical person. This means the brain reconfigured the visual map of the left hemisphere to express twice the visual space it originally would. When one door closes, another opens succinctly summarizes this remarkable process. However, if blindness occurs in adulthood, the adjustment and adaptability of brain maps will be limited because adults don't undergo the revolutionary restructuring of brain maps that infants do. For example, research has shown that individuals who play the piano have larger areas in their brain's motor map corresponding to both hands compared to those who don't play the piano. Furthermore, individuals who start learning to play the piano at a young age have even larger brain map areas corresponding to both hands than those who begin learning as adults. This is because brain maps in childhood are more adaptable, allowing for more flexible adjustments in function. Of course, adults have significantly stronger cognitive abilities than children, but this is because adults have developed new abilities by bridging cognitive gaps on the foundation of existing capabilities through a combination of multiple brain maps. However, the ability to bridge these cognitive gaps largely depends on childhood experiences, as the basis for developing new abilities in adulthood is still the fundamental brain maps established during early life. In this light, early life experiences have a profound impact on brain maps. After understanding this, how can we make brain maps serve us better? In recent decades, there has been increasing recognition that the prenatal environment and early life experiences have powerful and long-lasting effects on a person's health and happiness. These experiences can even shape our lifelong physiological functions, a fact strongly supported by animal experiments. For example, newborn mice are born blind and unable to walk. In response, mother mice often lick and comfort their offspring. The frequency with which the newborn mice are licked within the first week of life directly determines their future physiological stress response. Compared to mice that were frequently licked by their mothers, those licked less often exhibit higher stress hormone levels as adults, are more easily frightened, and have more difficulty adapting to new environments. These effects persist throughout their lives. In simple terms, early experiences reshape their brains and bodies, preparing them to be constantly vigilant against uncertainty and danger. The same holds true for humans. One of the most famous examples is the Netherlands during World War II, which experienced a widespread famine period during the Nazi occupation. Although the famine didn't last long, and the Dutch people enjoyed good lives after the war, that winter of famine left invisible scars on the children who were conceived or born during that time. Maternal hunger affected the prenatal environment, leading these children to develop metabolic disturbances related to type 2 diabetes and obesity in later life. These examples demonstrate that experiences during our days and childhood can influence our bodies and alter our lifelong physiological functions. Childhood is a period of intense learning and preparation, a time when the body and brain are frantically learning from the environment, and this period will indelibly shape your future. All we can do is fully recognize the value of childhood and strive to provide children with rich and interactive living environments to help them form various maps. These neural foundations will facilitate their lives throughout their lifespan. Considering the long-term effects of childhood experiences, protecting and supporting children is not only a moral obligation but also a wise investment in society. Additionally, we can provide extra knowledge and support for adults who didn't have rich childhood environments. While the flexibility of brain maps decreases significantly in adulthood, it doesn't disappear entirely. Even as adults, we can continuously improve our brain maps through effort. Therefore, helping these adults progress in society still holds significant benefits. Alright, we've covered most of the content of this book, so let's summarize briefly. First, we discuss the purpose and significance of brain maps. The brain needs to be smart enough to deal with external challenges, but due to resource limitations, it can't grow infinitely. This is where brain maps come into play. Their role is to help the brain focus its efforts efficiently. Brain maps enhance efficiency by generating maps, distorting them to prioritize important information, and using attention to help people concentrate on challenges. The importance of brain maps lies in the fact that they not only provide us with basic thinking tools but also enable us to flexibly assemble these tools, thereby gaining more complex thinking abilities. In this sense, human intelligence is rooted in brain maps. Next, we introduce the development process of brain maps and how to make them serve us better. The development of brain maps mainly consists of two steps: building a framework based on genes and then reshaping it based on environmental cues. Early life experiences have a significant impact on the development of brain maps. Children can flexibly adjust the distribution of brain maps, while adults have limited learning potential. Therefore, we should emphasize the early development of brain maps and strive to provide children with rich and interactive living environments to help them form various maps, preparing them better for life's challenges. Finally, in this last part, I'd like to share some future prospects for the application of brain maps. These applications give us hope for the future of brain maps. For those in a vegetative state due to illness or accidents, who may still retain consciousness but cannot communicate with the outside world, brain maps could be the breakthrough. The brain maps of different individuals show similarities when engaged in similar conscious activities. So, theoretically, a person with no physical response but with consciousness intact could convey specific thoughts mentally, which could be detected through technological means. Researchers at the University of Cambridge conducted a study on patients in a functional locked-in state. They placed these patients in functional magnetic resonance imaging, fMRI, scanners and asked them yes or no questions like, "Is your father's name Thomas? Do you have siblings?" If the answer was yes, they instructed the patient to imagine playing tennis. If the answer was no, they asked the patient to imagine walking around their home. Astonishingly, they successfully communicated with these patients by detecting different patterns of activity in the motor maps. While this technology is far from practical application, it does offer hope. Those completely trapped in their bodies, unable to communicate with the outside world, might one day regain the ability to express themselves through this mind-reading technique. An advanced version of this technique would involve controlling external objects through brain maps, which is highly attractive to many people with disabilities. In one study, scientists implanted electrodes in the right-hand area of a high-level quadriplegic patient's motor map and installed electrical stimulators in the muscles of his right hand, arm, and shoulder. A computer read electrical signals from the patient's brain map and converted them into movement commands sent to the electrical stimulators, enabling the patient to control the movement of his arm based on his thoughts. Once this technology becomes more widespread in the future, it will undoubtedly be a godsend for countless individuals with disabilities. Alright, that's the essence of this book. Congratulations on completing another book. Thank you all for your support and attention. Please subscribe to the Tim Booker channel, like this video, and share this valuable knowledge with your friends. Let's combine wisdom and practice to achieve our financial goals and create a better future. Thank you, everyone, and goodbye.